Dear viewers, greetings. In this video, we are going to see about the gram-positive coccobacilli, Listeria monocytogenes. First, general characteristics of Listeria monocytogenes. Listeria monocytogenes is a gram-positive coccobacilli arranged in form of chains. It is motile in nature due to the presence of peritracheous flagella and it shows tumbling motility. These Listeria monocytogenes are motile at 25 degrees Celsius and non-motile at 37 degrees Celsius. And its size is ranging from 0.5 to 1 micrometer in diameter, and it belongs to the family Listeriaceae. Capsules absent and endospores also absent. Regarding the mode of respiration, aerobic or facultative anaerobic mode of respiration is observed in Listeria monocytogenes. Optimum temperature for the cultivation of Listeria monocytogenes is 35 degrees Celsius to 37 degrees Celsius. It also able to grow at refrigerated temperature 4 degrees Celsius. The optimum pH of Listeria monocytogenes is 4.5 to 9.6. And regarding the habitat, Listeria species are mainly found in the soil. Though Listeria monocytogenes, a pathogen, may specifically be found in raw foods such as unpasteurized fluid milk, raw vegetables, raw and cooked poultry. Next discovery. The Listeria monocytogenes was discovered by Everett George John Murray in the year 1924 based on 6 cases of sudden death in young rabbits. Murray referred to the organisms as bacteria monocytogenes before James Hutner Harvey Peary changed the genus name to Listeria monocytogenes in 1940. Disease transmission of Listeria monocytogenes. Listeriosis infection in humans occurs through the consumption of contaminated foods, particularly unpasteurized milk, soft cheese, vegetables and prepared meat products. Unlike most foodborne pathogens, Listeria multiplies readily in refrigerated foods that have been contaminated. Transmission is also possible from mother to fetus or from mother to child during the birth. Some of the common virulence factors of the Listeria monocytogenes are cell attachment factors or internalins, hemolysins like toxin listerolysine O and enzyme phospholipase C, and protein that mediates actin-directed intracellular motility, that is ACTA. These three agents are the virulence factors responsible for causing the pathogenicity or disease in the host cells or human cells. The term virulence represents the ability of the microorganisms to cause the disease. The incubation period for Listeria monocytogenes infection is 1 to 90 days. Once the patient infected with Listeria monocytogenes, it can take from 1 to over 90 days for illness to develop. And the average incubation period is about 30 days and the infective dose is ranging from 107 to 109 CFU is the infective dose. Pathogenesis of Listeria monocytogenes. Listeria monocytogenes causes the disease called listeriosis. After ingestion of the contaminated food, Listeria monocytogenes is able to survive exposure to the proteolytic enzymes, stomach acids and bile salts through the protective action of stress response genes. The bacteria are then able to adhere to the host cells via the interaction of proteins on the surface of the bacteria, that is interleukin A, with glycoprotein receptors on the host cell surface, example epithelial keratin. And then infection is initiated in the M cells or microfold cells in Bayer patches in the small intestine and mucosal associated lymphoid tissue or MALT. After penetration into the cells, the acid pH of the phagolysosome that surrounds the bacteria activates a bacterial pore forming cytolysine or listerolysine O and two different phospholipase C enzymes, leading to release of the bacteria into the cell cytosol. The bacteria proceeds to replicate and then move to the cell membrane. This movement is mediated by bacterial actin protein or ACTA proteins. After movement to the cell membrane, a protrusion or phylopod, a long thin filamentous pseudopodium is formed, pushing the bacterium into the adjacent cells. After the adjacent cell ingests the bacterium, the process of phagolysosomes lysis, bacterial replication and directional movement repeats, and the entry into the macrophages after passage through a intestinal lining carries the bacteria to the liver and spleen, and finally it leading to the disseminated diseases. The list of clinical diseases caused by the Listeria monocytogenes are neonatal listeriosis in pregnant women, listeriosis in healthy adult, meningitis in adults, and finally primary bacteremia. The first clinical disease caused by the Listeria monocytogenes is neonatal listeriosis. Two forms of neonatal disease have been described. They are early onset disease and late onset disease. The early onset disease was acquired transplacentally in utero. Early onset disease can result in abortion, stillbirth or premature birth. Granulomatosis infantis septica is a severe form of early listeriosis which was characterized by the formation of abscess and granulomas in multiple organs, and a high mortality rate will be observed unless treated promptly. Next, late onset disease. Late onset disease occurred at or soon after the birth, and late onset disease occurs 2 to 3 weeks after birth in the form of meningitis or meningoencephalitis with septicemia. And the second clinical disease caused by the Listeria monocytogenes is listeriosis in pregnant women. Most infections in pregnant women occurs during the third trimester when cellular immunity is most weak. Pregnant women typically experience only fever and other non-specific symptoms, and the infection during pregnancy can lead to miscarriage, premature delivery or life-threatening infection of the newborn. The third clinical disease caused by the Listeria monocytogenes is listeriosis in healthy adults. Most Listeria infection in healthy adults are asymptomatic or occurs in the form of a mild influenza-like illness. An acute self-limited gastroenteritis developed in some patients which was characterized by a watery diarrhea, fever, nausea, headache, myalgia and arthralgia. Listeriosis in patient with compromised cellular immunity is more severe. The fourth clinical disease caused by the Listeria monocytogenes is meningitis in adults. Meningitis is the most common form of disseminated Listeria infection in adults. Although the clinical signs and symptoms of meningitis caused by these organisms are not specific, Listeria should always be susceptible in patients with organ transplants or cancer, and in pregnant women in whom meningitis develops. Disease is associated with a high mortal rate of 20 to 50%, and significant neurological sequelae, that is the complications involved in central nervous system, among the survivors is observed. The final and fifth clinical disease caused by the Listeria monocytogenes is primary bacteremia. A patient with bacteremia may have an unremarkable history of chills and fever. This kind of chills and fevers commonly observed in the pregnant women, or a more acute presentation with high-grade fever and hypotension or low blood pressure. Only severely immunocompromised patient and the infants of pregnant women with sepsis appears to be a risk of death. Next, laboratory diagnosis of Listeria monocytogenes. The first diagnostic method is microscopic examination. Under gram staining, the gram staining preparations of cerebrospinal fluid typically shows no organism because the bacteria are generally present in the concentrations below the limit of detection. If the gram stain shows organisms, they are gram positive coccobacilli. In the hanging drop method or motility test, these Listeria monocytogenes are motile only at room temperature, and at 37 degrees Celsius, these Listeria monocytogenes are non-motile in nature. And next, colony morphology in culture medium. In nutrient agar, Listeria monocytogenes grows as small round colonies observed on agar medium after incubation for 1 to 2 days. It may be necessary to use selective medium and cold enrichment technique to detect the Listeria in specimens contaminated with rapidly growing bacteria. Cold enrichment means storage of specimen in the refrigerator for a prolonged period. In blood agar, Listeria monocytogenes are beta hemolytic in nature, and in McConkey agar, non-lactose fermenting colonies were observed. Next, biochemical test. Catalase test positive, oxidase test negative, urease test negative, indole test positive, methyl red test negative, Voges-Prescott test or VP test positive, citrate utilization test negative, CAMP test positive, esculin hydrolysis test positive. And next, the molecular analysis of Listeria monocytogenes. Two techniques are involved in the molecular analysis of Listeria monocytogenes. They are PFGE or pulsed field gel electrophoresis and PCR or polymerase chain reaction. Finally, treatment and prevention of Listeria monocytogenes infection. The treatment of choice for severe disease is penicillin or ampicillin alone or in combination with gentamicin. People at high risk should avoid eating raw or partially cooked foods of animals' origin, soft cheese and unwashed raw vegetables. Dear viewers, that's all about the bacteria Listeria monocytogenes. Thank you for your support. Thank you.